Hello, it's me again, Alejandra. I haven't had any time to do any videos in the last couple of days, but I've learned so much about new conditions related to our blood and our bone marrow. And I wanted to go more in depth with the conditions, but I don't know, I just couldn't get to a point where I could explain properly before going through the basics first. So this video is going to be very short and it's just going to be super basic just to build a baseline for the following videos and the following conditions. A lot of information that I'm going to share, I found it online, but also I found this awesome book in my library, my home library. Um, it's called Hematopoietic Cell Transplantation. It's a second edition, edited by Donald Thomas, Carl Bloom, Stephen Foreman. It's a very old book. It was they actually almost threw it to the trash a long time ago, and I saw that and I asked if I could have it. I didn't know I was gonna use it. This was many years ago, and now look at me, I'm in hematology oncology, and it's super helpful. And although we have YouTube and Google and up to date, I just love the feeling of opening the book and reading, and I can highlight and I can tag it. So I'm enjoying that a lot. But, anyways, I wanted to just talk about something super basic, which is plasma cells. So, plasma cells are found in our bone marrow. They're part of our immune system and they produce antibodies or immunoglobulins, which are the IgGs that I've talked before in other videos. And these IgGs, immunoglobulins or antibodies, help our body fight infection. So I found this cool graph online, kind of to so everybody can see how the plasma cells are formed. For those that have taken microbiology classes or cell biology, you've probably seen many other versions of this graph, but pretty much that's the blood stem cell, and it can differentiate into myeloid stem cells or lymphoid stem cells, and then the lymphoid stem cells that are on the right differentiate into lymphoblast, and those can differentiate in three different categories: B lymphocytes, T lymphocytes, or natural killer cells, and the B lymphocytes also differentiate into plasma cells, which is what we're going to talk about. And plasma cells are part of our white blood cells in our bone marrow, and their main focus is to produce antibodies and help our body fight infection. So let's talk more about those. So like I said before, plasma cells produce antibodies or immunoglobulins. Here is a picture of our immunoglobulin. Immunoglobulins, there's different kinds, like the IgGs, the IgMs, the IgEs, but pretty much this is the structure. So they have the heavy chains are actually the immunoglobulins, and the light chains are um, either could be kappa or lambda chains. They're called the light chains and they attach to the immunoglobulins. And this is very important to understand about these light chains, kappa and lambda, because they have an important significance when we are ruling out different blood conditions. We usually check for the ratios between kappa and lambda, and that can help us determine what kind of condition a patient has. Okay, so this is a better graph of the heavy chains, which are the immunoglobulins, and the light chains, the kappa and lambda. So the important thing is that, for example, in myeloma, these plasma cells that are normally produced to fight infection become abnormal, and they start multiplying without control, and they produce large amounts of single type antibodies, which are called paraproteins or M proteins, that are usually not functional, but also these light chains that are present in the blood. Usually kappa and lambda are in a perfect or in a very close ratio, so there's the same amount of kappa that there's lambda. But we as hematologists check the kappa lambda ratio. If the ratio is low or high, meaning there's more lambda or more kappa, that is a big red flag for us to think, to start exploring and seeing if there's another problem in the bone marrow, because there should be a 1-to-1 ratio or close to that. When there's a below 1 or above 1 ratio, um, we worry that perhaps the patient has myeloma, that there's not the right balance of production of these immunoglobulins and light chains in the blood. And also, like I said before, we also check for proteins, which are uh, paraproteins and proteins, which are a different thing that I'm going to explain in a future video, but they are also abnormal things. But the thing that they're all connected is that they are all part of the plasma cells. So this is plasma cells that are acting abnormally by having different ratios of kappa and lambda. Okay. Okay, so now that we understand plasma cells better, and how they produce these immunoglobulins with the light chains kappa lambda. This is a great introduction to move forward to see these three different stages in which plasma cells are acting abnormally. So the first one is MGUS, which is monoclonal gammopathy of undetermined significance. We usually there's no symptoms, fewer signs of harmful plasma cells and protein in the blood. Then we have smoldering multiple myeloma. This is when the plasma cells are a little bit more abnormal, still no symptoms and more signs of harm of plasma cells and proteins in the blood. So under the microscope, we see more abnormal plasma cells. Perhaps the kappa lambda ratio starts being low or high at this point, which is a red flag that this is getting worse. And then we have full-blown multiple myeloma, which there's symptoms throughout the body, and the blood is full of cancerous plasma cells and proteins. So in the next video, I'm going to talk a little bit more in depth of how to detect MGUS or SMM or MM, what tests do we do, what ratios, what the kappa lambda ratio, how do we treat if we see these conditions. I hope this is helpful. I know it's very quick, but I just felt like we had to give this introduction, and I'll see you in the next video. Bye bye.